I hope you can see my slide now.
We can see.
All right, thank you, colleagues. We are looking at taste and smell today, and maybe you can, if you like, you can call it chemosensory perception, because chemosensory perception focuses on taste and smell. You can add to this chemothesis as well, which is one of the perceptions that involve taste and smell, but also in addition to that, some tactile sensations. So that's what we're going to focus on today. And just by way of introduction, in this season, especially this part of the world, I'm sure most of us are enjoying some lemons because of flu, because of COVID-19. We believe that lemon juice and even the pills can help to alleviate the symptoms of flu. So when you test such foods, we know obviously that there is some sour sensations and sometimes a bitter sensation. That's part of taste. And on the other side, you see, I did put some samples which I took from my master's thesis. These are deep fat fried orange flaked sweet potato crisp. Some of our panelists described them as, in terms of aroma, as having a butternut aroma. So already you see that people can perceive certain aromas, they can perceive certain tastes when they are interacting with food. So this is a very important topic, generally in terms of nutrition, in terms of supporting health, but also particularly in the InnoFoodAfrica project. This is a very important topic because we are trying to develop plant-based novel foods that can promote health and well-being. And us knowing that most of the plant-based foods may tend to have some bitter characteristics, some astringent characteristics, some sourness, the topic becomes very important. So what we want to do today is to try and understand what it is really about taste, what it is really about smell, that we need to come to terms with. So why do we smell and taste? Now, these are part of an evolutionary process in human beings and perhaps in other animals as well. They help us to safeguard our behavior. They help us to guide our behavior to where we can have good food and away from where we can have bad food. In terms of for example, food that can be poisonous to us, food that can be toxic to us, or food that can give us nutrition, food that is energy, food that is proteins, food that is minerals. So that is part of the importance of smelling and tasting. These senses also provide us with pleasure. I think you all know that part of the reason why we eat food, apart from nutrition, is also enjoyment of the eating experience. Because if an eating experience is not pleasurable, obviously people will not want to eat that food. They would rather find food that they can enjoy whilst they are eating. So taste and smell and other senses as well, they inform the hypothalamus about what is happening. In other ways, they help us to our body systems to trigger digestion, to start the digestion process, to feel hungry. In other words, when at some point you must take food, but for you to know that now you need food, you need to uptake nutrients, there must be some cues that come from within, and that is the purpose of taste and smell via the hypothalamus, so that we know now we are feeling hungry and you need to eat. Now I'm feeling thirsty and I need to drink something. And that is important from a physiological standpoint, because it means you can maintain your body system in balance, in good balance. So now the hypothalamus has many roles. It has many functions. Some of them are listed here. It controls hunger, it controls thirst, it controls body temperature, parenting, shivering, like when it is cold, it controls blood pressure. So it is actually a very important part of the brain that also controls taste, that also controls smells, and regulates how we behave according to the tastes and smells that we will have received from the environment. So our senses collect information for the hypothalamus to use, like sensors for an instrument. So all the five senses, we have the sense of smell, we have the sense of touch, taste, vision, and hearing. And you see that here I have encircled the nose and the mouth because they focus on the sense of taste and the sense of smell. So they collect information when we are interacting with food, when we are eating food. The lips, the tongue, the olfactory system in the nose, they collect the information about the odorants in the food. And then they help us, they send the information to the brain, specifically to the hypothalamus, which now decides what type of food is it, what type of smell is it the smell that we can consume safely, or food that we can consume safely, or is it the taste of the food that can be poisonous to you? So before you even proceed with digestion, you know already, because the hypothalamus has already interpreted that information. So that is how important the hypothalamus is. Now this is just a joke to say, if senses are scaled according to importance, we would somehow look like this. You can see here that the hands, they're very big, then involved in terms of touching, the sense of texture, sense of feeling, I mean. And if you look at the mouth, the lips, the tongue, and the nose, again, you see that especially the mouth region is very big, because there's sense of taste there, there's sense of texture there, and things like that. But that's not how things are in reality. So what happens really when we sense, when we smell foods, or when we taste foods? We know that in the food there are some testants, those chemical compounds that produce a certain taste. It could be sugar, it could be bitterness, it could be caffeine, it could be some phenolics. In terms of aroma compounds, it could be sweet, smelly aromas. So what happens is, when they reach the sense organ, they cause some stimulation at the sense organ, and the stimulus here is actually the smell or the taste in the food. When it reaches a certain threshold, a certain level, it causes what we call an action potential. In other words, it causes us to perceive something. But when it does not reach a certain level, that's why you see here is failed initiations, which means we do not perceive anything, because we have not reached a certain minimum threshold to cause what we call an action potential to be propagated in the neurons that send information from the lips, the tongue, the olfactory system, that is the chemical senses, to the brain, specifically to the hypothalamus for interpretation of that specific uh, taste or smell in the food. So that's what we mean about sensory activation. What happens for us to end up experiencing a certain smell sensation or taste sensation? Now, in terms of sensory learning, these pictures show us what happens. The statement says learning in the brain is making traces between nerve cells, and the traces grow stronger with repeated experience. So the information that is sent via the neurons to the brain, it could be smell information, it could be taste information. The more it is repeated, the more we are exposed to those smells in the food. In other words, the more the same food is given to us, and we get associated with us. Literature says that in the brain, there is a certain wiring system that develops such that the next time you are presented with, sorry for that, the next time you are presented with such foods, it's easier for you to recognize what the food are, to identify what the food are. So it works again, not only in terms of neurons, but also in terms of hormones. There are certain hormones that are produced associated with certain behaviors and certain foods. In this case, dopamine is produced by the pituitary gland, and it's a pleasure hormone. It creates pleasure, which means if you eat foods that promote or that have precursors for dopamine, there is a possibility of experiencing pleasure. That is what part of some of the research coming nowadays is pointing to. And the pleasure is associated with reward. In other words, to say this is good. Reward is to say this is something that is good and this is something that can be repeated, as opposed to punishment, where we say this is something that is not good and you may not repeat, or you may not want to eat that food again. So another hormone in that regard is serotonin, which is produced in association, or which influences mood, and is the precursors are actually ingested in the diet, not the hormone itself, but the precursors are ingested in the diet. So that is important, because it means you can actually choose or design your diet in a manner to include the foods that may influence specific mood behaviors, specific sensation behaviors. Now, nowadays, because of brain imaging techniques, it is possible to see some of these stimulations in the brain. For example, here, there is no serotonin, which is a hormone. Then a brain scan shows a depressed condition, someone who is not feeling good. Only a few parts of the brain are activated. Whereas when there is serotonin in the diet or in the blood system, the person is not depressed. In other words, the mood is jovial, the mood is good, because there is serotonin, and the brain scan shows that several regions of the brain are activated. So it is postulated that foods like eggs, cheese, nuts, amongst others, may be associated with stimulating mood, good mood. And there are precursors for melatonin, which is also a hormone that helps, that is produced by the pituitary gland and sent to the skin to help in the production of the skin pigment melanin, which prevents, which protects us against the ultraviolet heat. But also it is known that it helps us to have good mood. So now the brain can communicate through several mechanisms. I think we've explained this. It can communicate through the nervous system, from the senses to parts of the brain. It can communicate through chemicals and hormones. So this is part of what we have already explained. And here we see that dopamine, these are the happy brain chemicals. In other words, they are hormones that help the brain to remain in a happy state. We have dopamine, we have serotonin, we have endorphin, we have oxytocin, sorry. And as we mentioned already, dopamine is a reward hormone. It causes happiness, and serotonin, it causes mood, good mood, which means when there's an imbalance of serotonin, you may have some mood disorders. And these suppress pain, which means when there is no pain, obviously we are likely to be in a happy state. They are the feel good hormones. So in general, what the slide is trying to show is that if we are able to identify foods that have precursors which promote the production and increase of these hormones in the body, it means that we can affect good mood in consumers. So, for example, when mice were given inhibitors for fat uptake, they compensated by eating more fat to get the same dopamine response. Remember, dopamine is associated with reward. So because dopamine was inhibited, and it is an indicator um, of sugar and fat, which means when we eat sugar, when we eat fat, there is a release of dopamine in the body, and that makes us happy. In other words, we are rewarded. So when the system does not detect sugar or fat, there is a tendency to want to eat more so that you can compensate for the sugar and fat that is not being detected in the body. So what it means in terms of sensory science is that if we can manipulate these hormones so that there is a perception that they are there in the blood system and our system can detect them, it means that we can actually down regulate the need for sugar, the need for fat in terms of consumption. So why do we have taste and smell? And this is what we've explained in the first slide. So basically, we know that we have 5 basic tastes: umami, sour, sweet, bitter, and salt. And of late, of course, there's the 6th test that is being postulated, which is the fatty test, but I'm not going to talk much about them. This is almost common knowledge. So the tests are important because they are indicators or proxies of different nutrients in the diet. For example, when we test the sweetness, we relate that to energy. We know that there is energy in the diet. It attracts organisms, it attracts human beings towards diet that provides energy. And when you test saltiness, it regulates the salinity in the cells. And we know that salinity is about how salty or the osmotic potential of the body. And that is very important because salt, among its other ions, affect uh, disease states like hypertension, which is blood pressure in general. So when you test sour, you detect unripeness. It means you may want to avoid such foods. For example, fruits that are sour, there's a tendency to want to avoid them because you know that they are not ripe. That is the indication. Yet, unfortunately, common knowledge, contemporary knowledge nowadays tells us that most of the foods that are plant-based, most of the foods that may have a sour perception may tend to be healthy. So when you test bitter, you associate that with poisons, which is unfortunate again. It was good in the past because there was need to avoid all foods that are bitter. But nowadays we know that some of the bitter foods, for example, foods containing phenolics, uh, tend to be healthy. And when you test umami, you know that it leads you to conclude the food has proteins. So in terms of smell, it is important that everything that we eat, the first thing that we do is we smell the food. Actually, not consciously, but it happens subconsciously because the smells are volatile. They reach your nose uh, without you being aware of it, and you can make a judgment whether the food is good for consumption or not. So that's how important smell is as a gatekeeper of what can be consumed and what cannot be consumed. And like I have explained in the slide on activation of the senses, there are some levels or concentrations of testants, in this case odorants, that cannot be detected. You cannot tell what the smell is. In some cases, you can smell something, but you cannot identify it is because of subliminal sensations or subliminal sensing. This is because either the concentration is very low, our systems are wired in such a manner that there is a certain level beyond which we start to detect certain smells or certain testings. That's we call the detection threshold. And in some cases, we have what we call the recognition threshold, where you can now recognize what the odors are in terms of their identity. So that's what happens in the brain. I'll not go into the details about it, but here it's just saying the hypothalamus sometimes can detect that there is an odorant, but cannot actually attach a label to what the odorant is in terms of its identity. So the pituitary gland is actually located just below the hypothalamus, and if you like, you would describe it as the chief implementation officer of the hypothalamus, because it is the one that implements the decisions made by the hypothalamus. Decisions are made in the brain in the hypothalamus, and then they are directed to the pituitary gland, which works via different hormones to effect what has been decided in terms of taste, in terms of smell, and sometimes in terms of other sensory modalities as well. So here we see that the pituitary gland produces hormones into the blood, different types of hormones, gonadotropic hormones, which affect the muscles. And when muscles are affected, obviously it means that there's a motor activity. You tend to move away from a bad odor, or you move towards a good odor and things like that. And there are different hormones that are produced. Some of them affect the kidney. This is antidiuretic hormone, oxytocin, as we can see, prolactin, which are affecting the breast, and different type of hormones are produced. But the general issue here is that the pituitary gland is working under the control of the hypothalamus, and it also controls functions associated with taste, functions associated with smell. Um, it's a complex diagram that I will not get into a lot of detail. But for example, the kidney controls the amount of salts in the blood, which obviously means that affects tastes, which means when we eat salty foods, we are likely to have more sodium in the blood, and there's likely to have a lot of secretion of salty urine from the kidney, just as an example. So the intake of food is essential, obviously for nutrition reasons. We've talked about it, but also for pleasure reasons, and sometimes for nutraceutical reasons, for therapeutic reasons. And we have talked about the serotonin precursors. And these foods that are shown here, they are suggested as foods that contain serotonin precursors, the compounds that can be used to produce the hormone serotonin. One of them being the amino acid tryptophan. And serotonin, it is a mood stabilizer. It stabilizes the mood. It gives good feelings of well-being and happiness, and it stimulates production of melatonin. I think we've talked about it, which is also related to sleep. And oxytocin is another hormone that is being associated with these foods that are shown here. Most of them are fruits, and some are legumes, as you can see there, and some are cheese, as you can see them. And this is a hormone, again, it's a feel-good hormone, as you can see. It is associated with reducing stress, it's associated with love, it's associated with reducing anger and anxiety. That's how important oxytocin is, and this is found in foods like these. And just for interest sake, it's also one of the hormones that is found in higher concentration during child labor or in lactating women. Now we want to move on to the issues in chemosensory perception, contemporary issues that are coming up in the area of taste and smell research. One example that I'm giving here is the interaction between fat and beta test modalities, which is very interesting and important, because what it means that if the interaction is positive, we can use fat to mask or to reduce the intensity of bitterness in plant-based foods. So in this research, which was done by these colleagues, they found that the inhibition of beta test following simultaneous administration of fat and beta testings. And I'll not get into the details, but this is the diagram that they used to explain their model of how this happens. We see that there are beta testings here at position 2. There are also beta testings here, but there is also fatty acids. And when they go across the cells, into the cells, this we call it the endoplasmic reticulum, there is interaction at subcellular level, which causes the release of some neurotransmitter substances. One of them is serotonin, which we said is a feel-good hormone. And instead of feeling bitterness, we end up feeling better or good sensations because of that interaction. So we need to remind each other again that in terms of taste and smell sensations, there is also an interplay of genetics, to say that genes have an influence. There are some people who cannot test bitter substances, some who cannot test sweet substances, some who have different levels of sensations towards different concentrations of the chemosensory stimulants. For example, in this paper, beta test genetics, the relationship to testing, liking, consumption, and health, the authors were trying to explain how different groups of people who test or who experience different bitterness levels may end up having different consumption behaviors, which ultimately can affect their health status, because what you eat tend to affect your health outcomes. But just by way of an overview, these are some of the factors influencing food choice, food consumption, and food intake. We have alluded to genetics and biology here, demographic factors, experience or previous exposure, education level, health status. For example, if you know that you are sugar diabetic, you may not take a lot of sugar in your diet. If you are hypertensive, you may not take sodium chloride in your diet, or you may reduce the amounts of these compounds in your diet. So this is just showing the interaction of the different factors that affect food choices and food intake. We see already that most of the lectures in our InnoFoodAfrica sensory training workshop are interacting with each other, as we have said before. And the factors, obviously, they operate at different levels, including the individual level, at the group or family level, at the micro level. By the micro level, we mean, for example, the environment level, and at the macro level, which is the systems level. So another issue in terms of taste and smell sensations is the aspect of chemosensory plasticity. Now, this is an aspect that is very important, because as we know that in some cases, taste and smell can be genetically determined. That does not mean that we no longer have the power to modify behavior, the power to change circumstances. That's where chemosensory plasticity comes in. It says that these senses, sense of smell, sense of taste, can be changed, can be influenced by training. They can be influenced by exposure, so that even the sensations or even the tastes, even the smells that were previously unliked or previously less liked by a particular individual or a particular group, in the end, after training, may get to be liked much better than before. So that's what chemosensory plasticity mean. To say that it's not a fixed state, it's something that can be challenged, it's something that can be influenced in a particular direction. So our senses are highly plastic, and they depend on training, and they depend on learning, they depend on previous experience, and we call that the genotype by environment interaction. So that's what we've been explaining, to say under certain environments, we can change the liking for sweetness, we can change the liking for bitter tasting foods, we can change the liking for astringent tasting foods if that is to happen. For example, if it is associated with foods that are considered healthy. So there are mechanisms for doing this, and one of them we have already explained is about experience dependent changes, which means the more you experience it, the more you are likely to get associated with that, and that is normally associated with the method of repeated exposure. And there's the aspect of basic gain control functions, where the system adjusts to environments with more or fewer odors. Again, this is associative learning, which means I'll give an example of sugars here, not odors, but tastings. If you repeatedly expose people to sugar, to beverages that have significantly reduced the concentration of sugar for a long time, it is assumed that they end up liking the lower levels of sugar compared to previous. The same, for example, in salty foods, be it emulsified meats. If you expose consumers for a longer time to reduced salt concentrations, they end up liking the reduced salt content in those foods. So that's how it works in terms of mechanisms of chemosensory plasticity. Now, this is a case study where the researchers were showing the effect of exposing, for example, infants through the milk from their mothers on their dietary preferences later in life. As you can see, the topic says the presence of chemosensory cues transmitted through the mother's milk increases the search and ingestion response toward bitter and sour solutions. So what happens in this case was the researchers used the rodents or rats. They took the baby rats, and then after 3 days, they had to feed them. So initially, they gave two groups of mothers. The first baby rats were fed milk where the mothers were exposed only to water. And then the other group, the mothers were exposed to lemons. In other words, through the milk, the baby rats were being exposed to lemon in one group, and then they were being exposed to water in the other group. So this is exposure to maternal diet, to lemon, which is associated with bitterness, and to water, obviously, which was a control, which is associated with what is normal, not bitterness. So these are the results. What they found is that in terms of, for example, now after exposing them to the mother's breast milk, they now expose them to an artificial nipple, which has different concentrations of these substances: quinine, citric acid, saline or salt, and water, now being exposed via the artificial nipple. What they found is that those baby rats which were exposed to the mother's lemon via milk, they had higher body weight when they measured compared to those exposed to the mother. Those which were exposed to citric acid, they had also higher body weight, but those which were exposed to saline and water, there was no significant difference. They also looked at the number of grasps to say how many times are the rats running to grasp the artificial nipple to drink. And again, the higher number was associated with babies that were previously exposed to lemon in terms of quinine and citric acid, because these are related to lemon. So compared to the water, which consistently yet lower levels in all the experiments. The total time of grasp, how long did the baby race take while it's grasping the artificial nipple, which means they're enjoying. The longer they continue to grasp the nipple and drink, they are enjoying. So you see that in the water situation, the values were very low. And it goes like that until the latence. The latence, they say this is the time lapse before the race rushed to grasp the artificial nipple. In other words, they took time to decide to say, let me eat. Whereas those that were exposed to lemons, they quickly went for the diet with quinine, for the diet with citric acid, because it's associated to some extent with lemon, and these compounds made them to remember quickly what they were exposed before through their mother's diet. So previous exposure to diet may increase the likelihood that similar diets may be appreciated in life. And that is important in terms of plant-based foods, because we mean that plant-based foods contain phenolics, they contain astringent chemicals, they contain beta chemicals. If children are exposed at an early stage, for example, through breast milk, through complementary foods, there is a tendency that we can affect their health throughout the life cycle or in the long term. So just an overview. Another example here is when you use training and exposure. It's just an overview of what I was explaining in the previous slide, to say the relationship between chemosensory, gene variance, perception, training, liking, and dietary intake. So you see that this is a complex interaction. It's not one factor determining the outcome. It's an interplay of factors. So we start with chemosensory receptor genetics, or the genes, and then they determine perception and sensitivity. But we are saying that through training and exposure, as we have seen in the previous slide, you can modify this, and therefore you can modify liking and preference for certain foods, and that can increase intake, for example, of foods that are bitter, for food that contain astringent compounds, and that therefore enhance good nutrition and good health. So the expectation is that people will acclimatize over time with foods with lower salt, with sugar or fat, eventually becoming more palatable or even preferred by training and by exposure. And research is saying that sometimes it takes about 8 to 12 weeks to achieve this. Now, in this example, the researchers wanted to understand sweet liking phenotypes and their implications for obesity. To say, in a population, how do people like sweet taste? So what they found is that there are those who like sweet taste very much. They are called extreme sweet likers. What happens is, as you increase the sucrose concentration, their liking also increases. And the next group is the medium sweet likers. As you increase the sweet taste and concentration, which is sucrose in this case, you see that their liking increases until it peaks at some stage, and then it falls down, which means when it gets sweeter and sweeter, they begin to dislike the food. And then the third group is made up of sweet dislikers. These are people who really like low sweetness levels, such that when you increase the sweet concentration, their liking goes down. So these are the three groups. But the implication again is, how can you modify this via training? How can you modify this via exposure? As you can see that this group is at very high risk of diet-related non-communicable diseases associated with increasing sweetness. So which means they need to be trained, or you need to recalibrate their sweet liking preferences so that they don't like much sugar. In other words, their diet will not change. They will continue to like highly sweet foods, which can be not good for their health. So what are the research and public health implications of this? We have already explained, is to say that we need to understand the interplay among different factors that affect taste, that affect smell, that affect chemothesis, so that we're able to manipulate them to produce diets that can be liked by people to enhance good health and to enhance well-being. For example, nowadays we know that there is a lot of talk about reducing the amount of sugar in diet, reducing the amount of salt in diet, reducing the amount of even fats in diet. To do that, we need to understand the distribution of liking preferences for these different test modalities, and even the detection threshold, even the recognition threshold, so that we know that if we are going to reduce in our formulation to this level, what are the implications? Are people going to accept the product and things like that? And also we need to understand the genetics of it, so that we know that there are certain levels that we can influence by modifications, and there are certain levels that we may not be able to change perhaps. And with this, I would like to say thank you. It's time for questions.
Hi, James. I don't know if you can see the chat box, but there's a question in the chat from Nomzamo Muganu.
Nomzamo, thank you very much. I see the question says, since dopamine release is limited to sugar and fat intake, do you foresee a possibility for somehow making people feel rewarded or happy upon eating indigenous, climate resilient, crop-based foods? Nomzamo, this is a very interesting question indeed. I wish you were tackling it yourself. That is the challenge actually for not only sensor and consumer scientists, but even for food product designers at different levels, because we want consumers to experience the reward experience, to feel pleasant after eating. Yet we are saying that most of the plant-based foods may contain phenolics, may taste bitter, and that does not support the dopamine experience. I'll continue to say, for now, perhaps we need to keep researching about the food additives that we can add to plant-based foods to make them, to ensure that the consumers still experience the sweetness, for example, but without contributing the negative aspects of, for example, sugar. We know that it is associated with a rapid increase in glucose levels. You see, for example, we know that salt is associated with hypertension. So one common example that comes to mind is the artificial sweeteners, and people, they are already being used in different foods. By adding artificial sweeteners, for example, to plant-based foods, I hope that can help. But also, we need to think about natural sweet testings that can be added to plants, because people again are getting concerned about chemical additives to plants, and that can be a concern. So it's an area that we all want to think about how best to deal with that, so that the dopamine experience, the reward experience, continues to be available. Thanks, Nomzamo, for the question. And I see that Riet has mentioned that orange flesh sweet potato has a sweet taste, and also color can subtly modulate expectations of sweet, and that's actually a very important concept which she's bringing in here. One, not all, and thank you for that again, Riet, because we tend to overemphasize the bitterness, the astringency, and overlook some of the naturally good chemosensory properties of plant-based foods. For example, orange fresh sweet potato, we know that is sweet, it's associated with sweetness, and also it has some appealing colors, the orange color, which actually attracts consumers to want to eat the food. All these are pleasurable experiences, and they contribute to reward. So we can look at those positives and try to enhance them in our formulation of plant-based foods. Thank you for that, Riet. I don't, if there are any other questions, please, let's have them. Yes, reward can also come in the form of satiety, to say you quickly experience a sense of fullness when you are eating the food. So that's another aspect of reward. There are so many aspects of rewarding the body system. Remember, all these things are psychological. They are happening in the mind. So if we can find ways that ensure that we experience reward and we minimize the punishment aspect, in other words, the negative sensations, then the better. Yes, Nomzamo is adding the aspect of education, knowing that food is health promoting. Otherwise, information about food. So we can actually go on an overdrive to educate consumers, to raise awareness that plant-based foods, in many cases, in many circumstances, they are good. They have health promoting properties. They can promote non-communicable diseases, and that helps them to go for them. They will not only focus on the negative aspects, but they will look at everything collectively. And when they weigh all the options, they will realize that plant-based foods are actually good for them. Thank you for that, Nomzamo. So with these colleagues, I think we can stop the recording and we join in the next lecture at, let's say, exactly 12 o'clock. Thank you so much.
Yeah, James, thank you for your wonderful lecture. It's good. Thank you.
Thank you so much. Thank you.